Hi everyone, and welcome to Chapter 8 on photosynthesis. In our last chapter, which was Chapter 7 on cellular respiration, we talked about how cells harvest energy from nutritional molecules, and we specifically focused on sugar. This chapter is going to be a continuation of that discussion as we now answer the question of where do organisms get the molecules from which they harvest energy in the first place? Because while every organism does undergo cellular respiration in order to convert chemical energy into ATP molecules, the source of the molecules that are broken down through cellular respiration can differ. Here on the slide, I have two different types of organisms. I have a plant and an animal. And each of these organisms gets its molecules from a different source. The plants, for example, are able to take the energy of sunlight and put it through a process that converts that sunlight energy into chemical energy in the form of those macromolecules, the lipids, carbohydrates, and proteins. Animals, on the other hand, do not do this, but rather they consume plants or other organisms, and that's where they get their source of the molecules that provide them with energy. We distinguish these two types of organisms into two different categories, and they have specific names: autotrophs and heterotrophs. So, autotrophs are organisms that are capable of harvesting the energy of sunlight and converting it into chemical potential energy for their own consumption. The term autotroph has the prefix auto in it, and auto means self. So, what this term really means is that these organisms are able to make energy for themselves. Heterotrophs, on the other hand, are not capable of this. Instead, they have to consume the chemical energy that is produced by autotrophs. And you may think to yourself, well, not all animals eat plants. Some animals are strict carnivores and they only eat other animals. And yes, that may be true. However, if we think back to that question that I asked you in the last lecture, the thought exercise of what did you eat for breakfast today? Every single one of the possibilities that I presented to you could be originally traced back to autotrophs. Let's start with something easy, like the fruit in the fruit and yogurt bowl. Fruit is obviously a plant, right? And plants gets their energy from the sun. But what about something like the bacon and eggs? Well, eggs are laid by chickens, and in order to live, chickens have to consume some sort of a grain material, and that grain material is a plant that gets its energy from the sun. What about the bacon? Well, pigs are omnivores, so they may eat a wide variety of things, but whatever they eat, it has to have obtained its energy from the sun. Regardless of what sort of animal you are talking about, even if it eats other animals, eventually you make your way down the food chain to a point where the animal eats plants, and those plants are autotrophs that get their energy from the sun. So, really, all of the nutritional macromolecules that you consume, these contain converted sunlight energy. You are eating sunlight energy every time you eat something, um, although it may be a little bit farther removed from when it was sunlight energy. It originally came from the sun. So, in the biological world, as we've talked about before, the ultimate source of energy is the sun, and that energy flows through the autotrophs into the biological world and up the different levels of heterotrophs, which are considered consumers of the autotrophs. So, to summarize, what we just talked about in Chapter 7 was this process called cellular respiration that takes nutritional molecules and converts them into molecules of ATP, which can be used to drive processes for an organism. Now, we're going to talk about how these molecules, these nutritional molecules like sugars, are made in the first place. And in that process, we are going to be reviewing the steps involved in photosynthesis, which is the making of chemical energy from sunlight. One thing that I want you guys to understand is that it is not a sort of dichotomy where plants perform photosynthesis and animals perform cellular respiration, but rather plants perform photosynthesis to make the sugars, and then they perform cellular respiration to break them down. Animals only perform cellular respiration, so they share that in common with plants, but what they do not have in common with plants is that they do not make the sugars themselves because they cannot perform photosynthesis. So here's a look at a short list of some of the types of organisms that are able to perform photosynthesis. There are many different types of photosynthetic bacteria called cyanobacteria, which are capable of obtaining energy from sunlight. There's also aquatic algae, which includes things like seaweed, as well as microscopic forms of algae that may grow like scum on a pond. And then finally, there are land plants, which are the ones that we are most familiar with. And as we move forward in this lecture, we are going to be talking about the version of photosynthesis that takes place inside of aquatic algae and land plants. This type of photosynthesis is a little bit different from the type of photosynthesis that is done by bacteria, because while bacteria do not have any internal structures, as they are prokaryotic cells, algae and land plants are both eukaryotic cells, and therefore they have internal compartments that specialize in performing certain processes. And in aquatic algae and land plants, the special compartment or organelle where photosynthesis takes place is called the chloroplast. This microscopy image shows a number of plant cells, and inside of them there are these little green spheres, and those green spheres are the chloroplasts themselves. It's an extremely important organelle for plants because it's how they get their energy. If we were to be able to zoom in even further on these individual little green blobs that are the chloroplasts, then here's what they might look like on the inside. So the chloroplast has two layers of membrane. If that sounds familiar, then it's because we're thinking also about the mitochondria from last chapter, which also had two layers of membrane, an outer layer and an inner layer. But within the inner layer of membrane, there is a region of gelatinous substance called the stroma, which is kind of like the cytoplasm of the cell at large. And then sitting in the stroma are these stacks of little green discs called thylakoids. The stacks themselves, which kind of look like stacks of pancakes, the stacks themselves are called granum, and the discs within each stack are called thylakoids. Just like with cellular respiration, we are going to be walking through the steps involved in photosynthesis, and as we are doing that, we are going to be aware of where each step is happening inside of the chloroplast. So keep this structure, this anatomy of a chloroplast in mind. Now with cellular respiration, we started off by taking a look at the big picture or the broad scope of the process, which included looking at the overall recipe in the form of this chemical equation here. So naturally, we're going to do the same thing for photosynthesis. What are the ingredients that go into the process of photosynthesis? Everything that you see on the left-hand side of this arrow right here are the reactants or the ingredients that are necessary for photosynthesis to run. First of all, you need energy, and that energy comes from sunlight. The plant also needs CO2 from its atmosphere, and this CO2 is actually going to be the molecule that gets put together and built into molecules of sugar. And then the plant needs water from its environment, which it takes up through its roots. And all of this gets converted into molecules of oxygen, which for photosynthesis is actually considered a waste product and is the reason why plants give off oxygen into their environment. And finally, a molecule of C6H12O6, which is glucose, that same sugar that we were talking about in the last chapter. And if something looks familiar about this equation, then it's probably because you've noticed that it is the exact opposite of the equation for cellular respiration. Whereas cellular respiration takes oxygen and glucose and breaks it down into carbon dioxide, water, and energy, photosynthesis takes energy, albeit in a different form, and carbon dioxide and water, and builds it up into glucose and releases oxygen. So the neat thing about these equations is that if you remember one of them, then you can remember the other one. And it's easy to remember which one belongs to photosynthesis versus cellular respiration because we know that photosynthesis for plants, they take in CO2 and they give off oxygen, whereas for consumers or heterotrophs, it is vice versa. In our last chapter, when we were talking about cellular respiration, we looked at the process of sugar molecules being broken down over the course of glycolysis, pyruvate oxidation, the citric acid cycle, and oxidative phosphorylation to be turned into ATP energy. Now, we're going to instead be looking at the process of building those sugars up in the first place from the energy of light. So we're going to walk through the stages in this process, and you'll be happy to know that whereas cellular respiration had 4 separate steps involved in it, photosynthesis only has 2. The 2 stages of photosynthesis are the light-dependent reactions and the Calvin cycle. The light-dependent reactions take place in the membrane of the thylakoid discs, whereas the Calvin cycle takes place inside of the stroma of the chloroplast. So both stages take place inside the chloroplast, they just happen in different locations. We're going to walk through these stages one by one, starting with the light-dependent reactions and then moving on to the Calvin cycle. And as you can imagine, based upon their name, the light-dependent reactions depend upon light. This is where the sunlight energy gets captured and turned into chemical energy. So before we can talk about the light-dependent reactions, we need to talk a little bit about the physics of light and how it moves and how it works. So any wave of light has a set of properties that is associated with it, a set of quantities that can be used to measure the energy in that wave. One of those quantities is the wavelength. The wavelength of a beam of light is the distance between 2 peaks or 2 troughs within that wave. Because the light is traveling as this undulating up and down beam, although you can't see that when you look at it, the wavelength is the distance between any 2 peaks and any 2 troughs. Another quantity that we can measure is called frequency, and we'll talk about frequency on the next slide. But it's important to note that light travels both in the form of these continuous waves as well as discrete particles that are called photons. So when we refer to the word photon, what we're really talking about is sort of this particle, this package of light waves that are hitting the plant. Now, as we said, wavelength is the distance between 2 peaks or 2 troughs. Frequency is a measurement of how often a peak or trough occurs. And these 2 quantities are inversely related to each other. What this means is that a higher wavelength inevitably results in a lower frequency. For example, we have 2 different kangaroos here on the slide that are hopping at different frequencies and with different wavelengths. The kangaroo on the top has a shorter wavelength because there is a shorter distance between 2 peaks and 2 troughs within this wave. This means, of course, that this kangaroo has a higher frequency associated with its waves, meaning that there's a higher number of waves that occur in a given distance. The kangaroo on the bottom, however, has a larger wavelength, but it has a lower frequency. So, key message here, wavelength and frequency are inversely related. Where one goes up, the other one inevitably goes down. They are also related to how much energy is being carried by a beam of light, because the lower the wavelength and the higher the frequency, the more energetic a beam of light is. And you can probably just see how there's some intuitive sense of this looking at the beams of light. Which one is more energetic, the kangaroo on the top or the kangaroo on the bottom? Obviously, it should be the kangaroo on the top, and this is the one that has a shorter wavelength and a higher frequency. So shorter wavelength and higher frequency equals higher energy in the beam of light. Now, this is not only true for light, but it's also true for all other types of energy that we see along what is called the electromagnetic spectrum. The electromagnetic spectrum shows the whole range of different types of energy that exist in the world, all the way from the lowest frequency and therefore lowest energy waves, such as radio waves, and then moving up through microwaves, infrared waves, the visible light spectrum that we are actually able to see, into ultraviolet, X-rays, and then finally the very high frequency and high energy gamma rays. So if we just focus on the little range of the spectrum that constitutes visible light, which is the type that is captured during photosynthesis, you tell me in this first checkpoint, what color of light is more energetic? Blue light or red light? Based upon its frequency and its wavelength, which color is more energetic? So now let's talk about how these waves of light are actually captured and turned into chemical energy. We previously discussed how within a molecule, it is the electrons that carry the energy in that molecule. And so in order to capture the energy of a photon of light, that energy has to be absorbed by the electrons in a molecule. When certain types of molecules are exposed to photons of light, their electrons undergo a process that is described as becoming excited. Excitation of electrons involves those electrons jumping from an orbital that is closer to the nucleus up to an orbital that is farther away from the nucleus. And this is because electrons that are farther from the nucleus store a larger amount of energy and are more reactive compared to the ones that are held in close quarters to the nucleus itself. So this is how energy of light can be captured by a physical molecule. The types of molecules that are capable of this activity are called pigment molecules. Not all molecules can absorb the energy of light and allow their electrons to become excited, but pigment molecules can. Pigment molecules have a specific construction to them, and the one that you're looking at here is one of the major pigment molecules that is found in plants called chlorophyll A. As you can see, it's a complex structure, but there are a few features that we can pick out, and one of them is that there is this long chain of carbon and hydrogen atoms here, which is referred to as the antenna of the pigment molecule. We can kind of think about the antenna as being like the lightning rod that sticks out and the electrons get excited at the end of the antenna, and then their energy is passed down the antenna back to the main portion of the molecule, which is called the reaction center. And the reaction center is where that energy ends up being concentrated. Pigment molecules in plant cells are embedded in the thylakoid discs that are found inside of the chloroplasts. Hence, the light-dependent reactions that we are about to talk about occur in the thylakoid discs because that's where the pigment molecules are found. Now, chlorophyll A is just one type of pigment molecule that is seen in plants. There are actually multiple different pigment molecules, and each pigment molecule has a specific wavelength of light that it does best at absorbing. So as we can see here, chlorophyll A, the molecule that we were just looking at, is really good at absorbing red wavelengths of light, and it also is really good at absorbing blue wavelengths of light. This is different for other pigments such as carotenoids or chlorophyll A's cousin, chlorophyll B. But if we look at this figure, this graph, which shows us some of the more common plant pigments, we can also see that there is a certain region of light that appears not to be absorbed by the plant pigments. Tell me, what color is this light that is not absorbed by the plant pigments? And then speculate for me, what happens to this light instead if its energy is not being captured? So now that we've reviewed the principles of light and how it gets captured, we can move on to talking about the topic at hand, which is photosynthesis. And as we mentioned, the whole process is divided into two separate stages, the first one being called the light-dependent reactions, which you see on the left, and the second one being called the Calvin cycle, which you see on the right. So we will start with phase one, the light-dependent reactions. The light-dependent reactions are responsible for capturing the kinetic energy of sun waves and converting it into these short-term temporary forms of energy, ATP and NADPH. Now you're familiar with ATP. We haven't heard the acronym NADPH before, but we have heard NADH. We saw in cellular respiration that NADH was an electron carrier that would pick up hydrogen atoms, including electrons and protons, from glucose and its derivatives, and deliver them to the final step of cellular respiration, oxidative phosphorylation, where they were converted into more ATP molecules. Well, in photosynthesis, this NADPH molecule performs the same sort of function of carrying electrons from one place to another, except instead of being called nicotinamide adenine diphosphate, it has an extra phosphate group in it, and that's where that P comes from. So it's just like NADH, except it's NADPH because it has an extra phosphate group. So, bottom line here is that the light-dependent reactions allow the cell to capture the energy of sunlight, and then that energy of sunlight is converted into ATP and NADPH. And on the next slide, we're going to see how that happens. And of course, this whole process is happening inside of the thylakoid membrane, which is the membrane surrounding those little discs that are stacked up like green pancakes. Here is what the light-dependent reaction series looks like. We have a series of proteins that are embedded in a membrane, and if this looks familiar, then it's probably because it looks a lot like the fourth and final step of cellular respiration, which is oxidative phosphorylation. And in many ways, photosynthesis is the exact reverse of cellular respiration, not only in the sense that their overall recipes are the exact opposite of each other, but as we will see, we start with something here that looks a lot like oxidative phosphorylation, and then we're going to move into a cycle, which is in many ways the opposite of the citric acid cycle, and then we'll move on from there. So, we've got a thylakoid disc right here. And this is the membrane of it. It's a phospholipid bilayer. Embedded in that membrane are a series of proteins. Two of these proteins in particular are very important, and they are called the photosystems. Specifically, photosystem 2 and photosystem 1, which I've highlighted here. And these proteins are especially important because they contain the pigment molecules, which are responsible for absorbing the energy of sunlight. You may notice that in the order of things, photosystem 2 comes first, whereas photosystem 1 comes second, and that's kind of counterintuitive. However, the photosystems were named in the order in which they were discovered, not in the order in which they are relevant to the process. So that's just an annoying little detail that you have to remember, that 2 comes first and 1 comes second. So let's take a look at what happens in the light-dependent reactions. In the very first step, photosystem 2 absorbs a photon of sunlight energy. And as it does this, the electrons in the pigment molecules are excited. Those electrons are then passed from photosystem 2 over to this little molecule right here called plastoquinone. Plastoquinone is a type of electron shuttle, which we also saw in oxidative phosphorylation. It's responsible for transferring electrons from photosystem 2 over to the B6F complex here. When photosystem 2 loses these electrons from its pigment molecule after they become excited, it has to replace them. And the molecule that it grabs onto to steal electrons from in order to replace the ones that it lost is a water molecule. This water molecule, of course, has been absorbed by the plant through its roots, and now it is used as a source of electrons to replace the ones that were excited by the photon of light energy. So this water molecule is split, and some of its electrons are stolen, and when it is split, the product is some of these hydrogen ions, these protons, as well as molecules of oxygen gas. This is where the oxygen gas comes from that is given off by plants and other autotrophs. It comes from the splitting of water molecules in order to replace the electrons that have been excited by sunlight and forced to leave photosystem 2. Let's continue to follow these electrons on their path. So once the electrons move to plastoquinone, plastoquinone shuttles them to the B6F complex right here, and it uses the energy of these electrons to pump protons, or in other words, hydrogen ions, from the stroma into the interior of this thylakoid disc. This should be familiar as well because in cellular respiration, we saw that the electrons, as they bounce through this electron transport chain, their energy was used to pump protons from one side of the membrane to the other. After that pumping action is complete, then the electrons are passed to another electron shuttle called plastocyanin. Plastocyanin will transfer the electrons from the B6F complex over to the next photosystem, which is photosystem 1. Now, one thing that we need to know is that by the time these electrons reach photosystem 1, they have very little free energy left. And so in order to re-energize those electrons, they need to be excited by a second photon. And that's what happens to them at photosystem 1. Another photon comes in, hits the photosystem, its pigments are excited, and those electrons become re-energized. The re-excited electrons are then passed from photosystem 1 over to another shuttle called ferredoxin, and that shuttle carries them to this enzyme here called NADPH reductase. The name of this enzyme is completely self-explanatory because it tells you that what the enzyme does is it reduces NADPH. In other words, it adds electrons to it. Remember, oxidation and reduction. Reduction means gaining electrons, oxidation means losing electrons. So these electrons, in the process of being transferred to an NADP molecule, engage in the reduction of it and therefore produces NADPH. So this NADPH is generated as one of the products of the light reactions, and we will see that this NADPH will come back into the picture in the Calvin cycle. The other thing that has been achieved over the course of this electron transport process is that a large number of protons or hydrogen ions have been concentrated inside of the thylakoid. And just like we saw in cellular respiration, those protons want to diffuse to an area where they can be more spread out. And they do this by moving through that enzyme that we talked about before called ATP synthase. The protons rush through this channel. As they do that, the enzyme spins like a water wheel, and it uses the energy of that rotational motion to recharge molecules of ADP back up into ATP. So now we're finished talking about the light-dependent reactions, and I want to pause and recap by asking you, what are the products of the light-dependent reactions? What comes out of this first step of photosynthesis? And to give you a clue, there are 3 things, and I want you to make sure you hit all of them. I also want you to, in this checkpoint, tell me which components of the overall equation for photosynthesis are either produced or consumed during the light-dependent reactions. Which components did we see active during the first stage? That completes our discussion about the first phase of photosynthesis, the light-dependent reactions. And now we're going to move on to the second phase of the process known as the Calvin cycle. So the Calvin cycle is going to pick up two of the products of the light-dependent reactions, which is the ATP and the NADPH molecules, which are the temporary energy carriers of that sunlight energy. And they're going to use those to convert into a more long-term form of energy storage, and that is molecules of sugar. The Calvin cycle is no longer occurring in the thylakoids like we saw the light-dependent reactions doing, but instead it is occurring in the stroma of the chloroplast, so the jelly-like substance that makes up the interior. Now, true to its name, the Calvin cycle is indeed a cycle. And so we're going to walk through the cycle step by step because it can be divided into three different stages. Stage 1 is called carbon fixation, stage 2 is called reduction, and stage 3 is called regeneration. And we can see that the very first stage up here, carbon fixation, involves taking in 3 molecules of carbon dioxide per run through the cycle, and that carbon dioxide, of course, is sourced from the plant's environment. This is why plants take in CO2, because they are using it as the raw material from which sugar is made, and by using the energy of sunlight coming from the light-dependent reactions, they will, through this step, build these molecules of CO2 into a sugar molecule. So the next time you bite into an apple or eat a vegetable, think about the fact that what you're eating actually used to be carbon dioxide. You're eating former air. So here's what this step of carbon fixation looks like. When the plant takes in carbon dioxide molecules, it needs to attach them to an existing molecule to start to build this carbon scaffold that will eventually become the molecule of glucose. The molecule that the carbon dioxides get attached to is called ribulose 1,5-bisphosphate, or simply RuBP. You only have to know it as RuBP. So the cell has a constant supply of RuBP because it's always being regenerated. And when the CO2 comes in, it will be attached to the RuBP. And you can see where the CO2 has entered and attached to the RuBP right here. And then immediately, that RuBP molecule that has picked up the CO2 gets split in half into 2 molecules of what is called 3-PGA or 3-phosphoglycerate. It's important to pay attention throughout this cycle to the number of carbon atoms that are present in each of the molecules that we're talking about. So RuBP contains 5 carbon atoms in it. CO2 contains 1. And so when the 5 carbon atom RuBP picks up the CO2, it becomes a 6 carbon atom molecule. When that 6 carbon molecule gets split in half, each of these 3 PGAs contains 3 carbon atoms in it. Now, this very first step that we just talked about, the carbon fixation step, is catalyzed by a special enzyme called Rubisco. And Rubisco is actually an abbreviation of the full name of this enzyme. The full name of the enzyme is ribulose 1,5-bisphosphate carboxylase oxygenase, but again, you don't have to know that. I want you to know Rubisco. Rubisco is thought to be the most abundant enzyme on Earth. And just a little while from now, we're going to talk about why that is the case. But you can start brainstorming on that right now. Why do you think Rubisco is the most abundant enzyme on Earth? So in the first step of carbon fixation, we saw CO2 brought in from the atmosphere and get attached to an existing molecule called RuBP and then split in half to make these molecules of 3-PGA. In stage 2, which is called reduction, this is where we are going to see the products of the light-dependent reactions come into play. The molecules of 3-PGA are going to be transformed each into a molecule of G3P. And to do this, it takes the addition of some energy from the ATP that was made in the light-dependent reactions, as well as the addition of the hydrogen atoms and the electrons that were collected by the NADPH molecules. Here's another look at what this reduction process looks like. So here is the 3-PGA molecule, and I wrote ×2 here because we want to make sure that we understand that for every carbon dioxide molecule that enters the cycle, there are actually 2 separate 3-PGA molecules that get produced, because after it is added to RuBP, the RuBP splits in half into the 3-PGAs. So from this point on, we're talking about everything happening twice. That 3-PGA will get converted into a molecule called 1,3-bisphosphoglycerate through the addition of ATP energy to it. And then it will also pick up those hydrogen atoms and electrons that were carried from the light-dependent reactions by NADPH. The result will be that the 3-PGA molecule is transformed into something called G3P, or glyceraldehyde 3-phosphate. And again, I just want you to know it as G3P. So now what we've accomplished is taken in molecules of CO2, attached them to a scaffold, split that scaffold in half to make 2 3-PGAs, and then each of those 3-PGAs was transformed into a molecule of G3P through the addition of energy and hydrogen atoms and electrons from the products of the light reactions. Now what's going to happen is the cycle is going to reach a branching point, because in order for the cycle to continue, the cell needs to have a constant supply of these RuBP molecules, because without RuBP, there is nowhere to place the CO2 that is incoming to make the molecules of glucose later. So actually, the cell has to channel almost all of the G3P that it makes back into the cycle in order to regenerate molecules of RuBP. That's why this step here is called regeneration. Specifically, for every 6 molecules of G3P that reach this point in the cycle, 5 out of those 6 have to go right back into the cycle and be regenerated and reorganized into molecules of RuBP using a little bit of ATP energy. Only 1 out of those 6 G3P molecules gets to exit the cycle and go on to become a glucose molecule. Before we talk about what happens to the 1 G3P that exits the cycle, let's take a look at what happens to those 5 molecules that re-enter the cycle and are dedicated to reproducing more RuBP. So G3P is a 3-carbon molecule here. We can see the 3 carbons, 1, 2, 3. RuBP, as we saw, is a 5-carbon molecule. And so they're not equal in terms of their carbon content, and carbon content is the limiting factor here. Therefore, it takes 5 molecules of G3P in order to regenerate a grand total of only 3 RuBPs. If we look at all of the carbon atoms that are in these G3Ps on this side of the arrow, there's 5 separate molecules. Each molecule has 3 carbon atoms. Therefore, there are 15 carbon atoms over here. Because RuBP has 5 carbon atoms, you can only make 3 RuBPs out of the 15 carbon atoms that are brought together in these 5 G3Ps. So 5 G3Ps are recycled into making only 3 new RuBPs. That's what happens to the vast majority of the G3P that reaches this point in the cycle. But as we said, 1 molecule gets to leave the cycle. One is siphoned off in order to make glucose. And it turns out that a G3P is precisely 1/2 of a glucose molecule. In order to make a full glucose molecule, you need to collect 2 G3Ps, or in other words, 2 full runs through the cycle. A full run through the cycle is considered to be the collection of 3 molecules of CO2. And the reason why that is the case is because 3 molecules of CO2 will result in the production of 6 molecules of 3-PGA. Each molecule of CO2 attaches to a molecule of RuBP, and then each of those 3 splits to make 6 molecules of 3-PGA. Each of those 6 becomes a molecule of G3P, and then out of those 6, 5 go back into the cycle to remake 3 molecules of RuBP, the amount that is needed to collect another 3 CO2, whereas one of them gets to leave the cycle. So therefore, it takes 3 molecules of CO2 to make enough G3P so that one molecule can leave the cycle and be siphoned off to make glucose. Remember that G3P is only 1/2 of a glucose molecule, and so therefore, in order to make a full glucose molecule, you need 2 molecules of G3P and 2 full runs through this cycle here. Knowing that, I want you to tell me how many carbon dioxide molecules must be fixed, must undergo carbon fixation, in order to produce not 1, not 2, but 3 complete molecules of glucose. How many times would you have to run the cycle to make 3 complete molecules of glucose? And how many CO2 molecules are fixed through each run in the cycle? In this checkpoint, just like we did in the light-dependent reactions, I want you to tell me which parts of the overall equation for photosynthesis did you see active in the Calvin cycle? So now let's get back to this special enzyme called Rubisco, which I mentioned just a few minutes earlier that was thought to be the most abundant enzyme on Earth. If you counted up all the enzymes on Earth, then Rubisco would be the most numerous one. Why do you think that is? Well, typically, the answer that people come up with is that there are a lot of plants on Earth, and this is an enzyme that is found in plants. And that is certainly true. There are a lot of plants on Earth, and that contributes to Rubisco being very abundant. But there's also another reason why Rubisco is the most abundant enzyme on Earth. Rubisco is actually a very inefficient enzyme. It is not very good at its job, and therefore plants need a lot of it in order to accomplish what they want to accomplish in photosynthesis. Just imagine if you ran a company and you had workers who worked very slow and very poorly, you would need more of those workers in order to accomplish the same amount of tasks than you would if you had a very efficient worker. So that's Rubisco in a nutshell. The reason why Rubisco is so inefficient and so bad at its job is because it actually technically has 2 jobs. The full name of Rubisco, as we saw before, was ribulose 1,5-bisphosphate carboxylase oxygenase. Now, ribulose 1,5-bisphosphate, that's the molecule RuBP. That is the molecule seen in the Calvin cycle that CO2 gets attached to in the first step of carbon fixation. But we can also see in the name of Rubisco that it is a carboxylase enzyme, but it's also an oxygenase enzyme, which means that it can either add a molecule of CO2 to RuBP, or it can add a molecule of O2 to RuBP. Carbon dioxide and oxygen gas. RuBP can take either one of those from Rubisco. So as a carboxylase, in its function as a carboxylase, Rubisco adds CO2 to RuBP in the process called carbon fixation that we just saw. As an oxygenase, Rubisco adds oxygen gas to RuBP. And this process is called photorespiration. So Rubisco is actually inefficient because it is existing at the crux of these two processes that are competing for its attention. Photorespiration is trying to get Rubisco to add oxygen to RuBP, whereas the Calvin cycle is trying to get Rubisco to add CO2 to RuBP. And Rubisco is not an intelligent enzyme. It will simply grab whatever molecules are in its vicinity and attach them to RuBP. So if there's more CO2 around, then it will primarily do its job in the Calvin cycle. On the other hand, if there's more O2 around, then it primarily will do photorespiration. Obviously, because photorespiration is pulling Rubisco away from the Calvin cycle, photorespiration decreases the efficiency of a plant's photosynthesis. And it turns out that this effect is even worse at higher temperatures. So at 25 degrees Celsius, which is somewhere around room temperature in Fahrenheit, for every 5 molecules of RuBP that Rubisco works with, 4 end up getting a carbon dioxide molecule, and 1 ends up getting an oxygen molecule. So at room temperature, essentially, this means that Rubisco loses 20% of its efficiency because while it focuses 80% of its attention, or 4 out of 5 RuBPs on the Calvin cycle, it focuses another 20% of its attention on attaching O2 to RuBP in photorespiration. And at higher temperatures, this gets worse because remember, Rubisco is not intelligent. It will just grab whatever is in its environment and attach it to RuBP. The reason why hot climates make the effect of this inefficiency worse is because plants get gases from their atmosphere through pores that are called stomata, which you can see in this microscopy image right here. These are little pores through which oxygen and carbon dioxide gas can pass. When it is hot outside, plants are capable of closing those stomata in their leaves, and they close them primarily to prevent evaporation of water. This is an adaptation that they have to prevent losing water in hot, arid environments. However, a side effect of closing the stomata is that it prevents more CO2 from entering the plant, and it also traps the oxygen that is inevitably being released from the light-dependent reactions. This means that inside of the leaf, there is a buildup of O2 and not enough CO2 around because there's not enough CO2 being brought in, the stomata are closed. Therefore, Rubisco is exposed to a lot more oxygen than it is exposed to CO2, and it starts to perform more photorespiration than it performs the Calvin cycle. These graphs here show the difference between the efficiency of Rubisco carboxylating, or in other words, adding CO2 to RuBP, versus oxygenating, or adding O2 to RuBP at various temperatures. And as we can see, as temperature increases along the x-axis here, less carboxylation starts to happen, but the oxygenation just continues to go up and up and up. And this, of course, is because the stomata are closed, which traps the carbon dioxide, or pardon me, traps the oxygen inside of the leaves and prevents more CO2 from getting in. So, because of the inefficiency that is presented by photorespiration competing with photosynthesis, plants that live in very hot and dry environments have evolved 2 different ways of adapting to this inefficiency. There are two groups of plants that have specific adaptations to photorespiration, and the first group is called C4 plants. C4 plants include a lot of different plants that are considered grasses. Important agricultural plants that are in this group include corn and sugarcane. And the C4 plants have solved their photorespiration problem by spreading out the steps in the Calvin cycle and collecting the CO2 in one type of cell and performing the Calvin cycle in another type of cell. This is the anatomy of a leaf that could be found in a C4 plant. And this specific type of anatomy is called the Kranz anatomy, which is the German word for wreath, and it's called the Kranz anatomy or the wreath anatomy because there are these types of cells called bundle sheath cells that are organized into little circles, as you can see here. Surrounding the bundle sheath cells are mesophyll cells, which I've also highlighted. And so what the C4 plants do is they first take in the CO2 in their stomata and they attach it to a 3-carbon organic molecule in the mesophyll cells, the ones that surround the bundle sheath cells. So we can see a stoma here. This is one of the pores in the leaf. The CO2 will come in here, and then in these mesophyll cells, it will be attached to a 3-carbon organic molecule, and the result will be the production of a 4-carbon molecule, which is why these are called C4 plants. The 4-carbon molecules will then be shuttled to the bundle sheath cells, where they will be very highly concentrated there. Now, there's not a lot of O2 in these cells, and by channeling all of the CO2 into the bundle sheath cells, these types of plants can make sure that there is a very high ratio of carbon dioxide compared to oxygen, which keeps Rubisco focused because it's not seeing a lot of oxygen gas and trying to perform photorespiration. Instead, it's seeing a lot of CO2 and trying to perform the Calvin cycle. The second group of plants that has a special adaptation for photorespiration and efficiency is called CAM plants. CAM stands for crassulucian acid metabolism, which you don't have to know, but it's named after the plant family Crassuluceae. Examples of plants that belong to this group are ones that we are familiar with living in the Sonoran Desert, such as cacti, succulents, and believe it or not, pineapple is in this group as well. CAM plants have a little bit of a different mechanism for making sure that they get the most for their money when it comes to photosynthesis. Because it's so hot in the CAM plant environment, what they do is they open their stomata, open the pores in their leaves only at night. So during night is when they collect the CO2, which enters their cells and is fixed to a 3-carbon organic molecule. During the day, when it is hottest outside and they are most at risk of losing water from evaporation, they shut their stomata, and then they release the CO2, which has been collected throughout the night, and channel it directly into the Calvin cycle so that, again, Rubisco can stay on track. It doesn't get confused by the fact that there's a lot of oxygen around because there isn't. There's a much higher ratio of CO2 than there is oxygen. So to summarize, the typical plant, which is called a C3 plant, runs the Calvin cycle and its carbon fixation process in the same type of cell, the mesophyll cell, and it does this both day and night. The C4 plant has what's called a spatial solution to the problem because it separates the area where the CO2 is collected from the area where the Calvin cycle actually takes place. The CO2 is collected in the mesophylls and then highly concentrated in the bundle sheath cells. And then finally, the CAM plants have what's called a temporal solution, meaning that it has to do with timing. During night is when they collect the CO2, and during the daytime is when they channel it into the Calvin cycle because they close their pores at that time, preventing more CO2 from getting in. And that wraps up our discussion of photosynthesis. So now we're going to finish off the lecture with a few checkpoints to test your knowledge. The first one here has 2 organisms on the slide, and for each of these 2 organisms, the beaver and the apple tree, I want you to tell me whether they are autotrophs or heterotrophs, first of all, and second of all, whether they perform cellular respiration, photosynthesis, or both. In this checkpoint, I want you to tell me whether photosystem 2, which absorbs light optimally at 680 nanometers, or photosystem 1, which absorbs light optimally at 700 nanometers, absorbs higher energy light. Which one is higher energy? The 680 nanometer wavelength light or the 700 nanometer wavelength light? And last but not least, fill in the blanks on this checkpoint. Some groups of plants have improved the efficiency of blank by reducing the rate of a competitive process known as blank, and they do this through evolutionary adaptations that maximize the amount of what and minimize the amount of what in cells that perform the Calvin cycle. When you're finished with this checkpoint, then you are finished with Chapter 8.